Biotechnology is the application of biology to the problem-solving and product-making processes. The most well-known application of biotechnology is the use of genetic engineering to produce therapeutic proteins and other medications. Since the earliest agricultural settlements, people have been utilizing biological processes to enhance their quality of life. Humans first used microorganisms' biological processes to manufacture bread, alcoholic beverages, cheese, and preserved dairy goods some 6,000 years ago. But today's definition of biotechnology, which was originally used to refer to the molecular and cellular technologies that started to develop in the 1960s and 1970s, does not include such activities. With the help of Genentech, a pharmaceutical business founded in 1976 by Robert A. Swanson and Herbert W. Boyer to market the recombinant DNA technology developed by Boyer, Paul Berg, and Stanley N. Cohen, a budding biotech sector started to form in the middle to late 1970s. Gene-engineered materials were initially produced by pioneering businesses including Genentech, Amgen, Biogen, Cetus, and Genix, largely for medical and environmental applications. The ability to create naturally occurring therapeutic compounds in greater quantities than could be obtained from conventional sources like plasma, animal organs, and human cadavers was the fundamental accomplishment of biotechnology in its early years. Additionally, recombinant proteins are less likely to contain infections or cause allergic reactions. Researchers in biotechnology are working to pinpoint the disease's fundamental biological origins and precisely intervene at that level. Producing therapeutic proteins to supplement the body's natural supply or to compensate for genetic deficits, as in the case of the first generation of biotech drugs, is one way to do this. Gene therapy, which involves inserting necessary protein-coding genes into a patient's body or cells, is a comparable strategy. Research towards the creation of conventional medications and monoclonal antibodies that halt the progression of disease has also increased in the biotechnology sector. One of the most significant developments in biotechnology during the last quarter of the 20th century was the efficient manufacture of monoclonal antibodies. By recognizing hitherto unidentified marker molecules on the surfaces of cells, sensitive tests for a wide variety of physiologically significant chemicals have been developed thanks to the specificity and abundance of monoclonal antibodies. The study of genes, genomics, the proteins they encode, proteomics, and the larger biological pathways in which they act allowed for such advancements. There are many uses for biotechnology, especially in agriculture and medicine. Examples include the application of stem cell research and cloning to replace deficient or dead cells and tissues, as well as the use of biotechnology to combine biological information with computer technology, bioinformatics, investigate the use of tiny instruments that can enter the human body, nanotechnology, and more, regenerative medicine. In an effort to synthesize upward from molecular biology toward chemical roots, tissues, and organs, businesses and university laboratories merge these disparate technologies. Biotechnology has been found to be useful in improving industrial processes through the discovery and production of biological enzymes that catalyze chemical reactions, catalysts, for environmental cleanup using enzymes that digest contaminants into harmless chemicals and then perish after consuming the available food supply, and in agricultural production through genetic engineering. Biotechnology's uses in agriculture have generated the most debate. Genetically modified organisms, GMOs, have received calls for bans from some activists and consumer advocacy groups, as well as for labeling regulations to alert people to the rising presence of GMOs in the food supply. Bovine somatotropin, BST, a growth hormone that increases milk output in dairy cows, was approved by the FDA in 1993, marking the beginning of the introduction of GMOs into agriculture in the United States. The FDA authorized the first genetically altered whole product the following year, a tomato with a longer shelf life. Numerous agricultural GMOs have since received regulatory approval in the United States, Europe, and other countries. These include crops that make their own insecticides and crops that can withstand the application of particular herbicides used to eradicate weeds. GMO foods have been found to be safe by studies by the United Nations, the U.S. National Academy of Sciences, the European Union, the American Medical Association, U.S. regulatory agencies, and other organizations. Skeptics, however, argue that it is still too early to determine the long-term health and ecological effects of such crops. The area of land planted with genetically modified crops expanded substantially in the late 20th and early 21st centuries, going from 1.7 million hectares, 4.2 million acres, in 1996 to 180 million hectares, 445 million acres, by 2014. About 90% of the maize, cotton, and soybeans that were grown in the United States in 2014 to 15 were genetically altered. The Americas were where the majority of genetically modified crops were cultivated. Quick biotechnology facts. Your body cells each contain 2 meters or so of DNA. It would span 200 billion kilometers if laid end to end. That would reach 1,333 times from Earth to the Sun. To put that into perspective, the same distance would be covered by sunlight in 7.4 days. Thanks to PCR, DNA fingerprinting, which is used for paternity testing or to identify offenders, may be done with the least amount of genetic material. Naturally, it can also be employed to establish innocence. The 300th prisoner in American history was released last year after DNA testing revealed he had been wrongfully convicted. The sources of newly emerging infectious illnesses are frequently found using PCR. During the SARS pandemic in 2002 and 2003, the technique was utilized to pinpoint the origins of the coronavirus and establish it as the cause, work that took just 1 year to complete. Without PCR, it would have been practically impossible to find a similar needle, a little virus, in a haystack, Earth. You are made up in part of genetic material that is not human-derived. Through a process known as horizontal gene transfer, or HGT, bacteria and viruses can introduce their DNA into your genome. Scientists have found that certain cancer cells exhibit HGT far more frequently than healthy cells, possibly leading to potential causes and treatments. The name information molecule for DNA is not a misnomer. 1 gram of DNA can hold up to 700 terabytes of data, and read-write DNA technology has also been developed. According to computational biologist Nick Goldman, if scaled appropriately, all of the knowledge in the world, including films, images, scientific papers, the internet, and similar materials, would fit in the back of a single van. Of fact, DNA's capacity to transfer genetic information from one cell, or cells, to another is what earned it the moniker of information molecule. That might seem evident now, but not for long. The first live cell on Earth without a living parent in 3.5 billion years was produced by researchers at the J. Craig Venter Institute in 2010 using a synthetic genome constructed from scratch, even though it was modeled after a living organism. However, the construction of the synthetic genome cost $40 million. The industry is confronting a bigger bottleneck than genome engineering. Since the Human Genome Project's completion in 2003, the world has improved its DNA sequencing capabilities, with a current capacity of over 1 trillion kilobases per year. Unfortunately, less than 230 million kilobases of DNA can be synthesized globally each year. Applications of genetic engineering and synthetic biology will be increasingly widely available as technology advances and costs decline in the coming years. Synthetic biology applications will also be greatly aided by other technologies. For instance, the company Autodesk, NASDAQ, ADSK, well known for creating AutoCAD and currently revolutionizing 3D printing platforms, is creating software design tools for programmable matter in living systems. Scientists will eventually be able to design and construct human tissues and organs, single-cell creatures with novel metabolic pathways, and programmable nanoparticles. Project Cyborg is currently only under beta testing. We have a long way to go before it is completely operational, but we must begin somewhere. What are a few of the practical uses of synthetic biology? Spiders are unable to feasibly mass-produce the immensely strong and flexible dragline silk because they are simply too territorial. By introducing the spider silk gene into more docile animals, such as goats, researchers at the University of Wyoming were able to remedy the issue. These unique dairy animals generate milk that contains several, extremely long strands of spider silk, which are subsequently extensively extracted from the milk using a spool. The goats are safe and content. When the research was revealed, the media went crazy with spider goat headlines, although harnessing the basic components of life, genes, to develop effective production techniques is nothing new. Chymosin was the first food enzyme created utilizing recombinant DNA technology in 1990. The chymosin gene from cows was cloned into bacteria and fungi, which can manufacture the enzyme during fermentation in far bigger amounts and of much higher quality than dairy animals' stomachs. In the US, engineered chymosin is used to make up to 90% of the hard cheese produced. That was for today. Thank you for staying with us. Don't forget to like our video and subscribe to our channel.